Hey guys, this is Tiffany Martin and this is Medical Microbiology, and we are working our way through Chapter 9, Controlling Microbial Growth in the Environment. So we've already done some terminology, we've already done how we evaluate and how we make some decisions on what type of microbial methods we're going to use, and now we're going to enter the portion where I'm going to talk about ways that we can control microbial growth in the environment. We're going to divide it into two big categories. We're going to talk about physical methods and then chemical methods. So physical methods is first, and this is just altering the environment. There's lots of different ways we can do this and not use any chemicals to control microbial growth in the environment. All right, so let's move into that. The biggest way we can control our environment and alter and control microbial growth in our environment is using heat. When we raise the temperature, we denature proteins, right? When proteins fall apart, the cell can't work. Cell membranes denature or get damaged with too much heat. So just by changing the temperature, and we're talking about environments, we're not talking about altering our temperature, we're talking about altering the environment. If we can change the environment, that'll control microbial growth. So heat, we do want to separate heat into two categories, moist heat versus dry heat. Let me ask you this question. So if I did truth or dare and I asked you, what would you rather do? If I gave you a dare, would you rather put your finger into a boiling pan of water on the stove for 1 second or even half a second? Or would you rather put your finger into an oven for a second? Which would be better? If you just put your finger in the oven or if you put your finger in the boiling water? Okay, I'm hoping you said oven. So if we put our finger in a boiling water, even for 1 second, we know that that can cause damage. If the boiling water splashes us, we know that that can immediately cause damage. But yet I can take my hands, and I'm using hot pads to hold the thing, but I can take my hands and stick it in a warm oven and be fine. And it's kind of interesting because water boils at 212 degrees Fahrenheit. I'm cooking chicken in my oven at 350 degrees Fahrenheit. That is hotter than my boiling water. What's the difference? The difference is, and you know this, is that water conducts heat very well. And so when I put my finger in the boiling water, that water is going to very quickly take that 212 degrees Fahrenheit and conduct a lot of that heat and burn my finger. In the oven, the air in the oven does not conduct heat very well. So even though the oven is hotter, 350 degrees, I can put my hand in as long as I'm, with the air surrounding it, and I'm okay. The air takes a lot longer to get my skin to a temperature that's going to cause some damage. Now we know all bets are off if we touch the side of the oven, the metal in the oven. Metal conducts heat very well, and I don't want to do that. If I touch a pan, a glass pan, a metal pan from that conducts heat a lot better. But there's a difference between moist heat and that water coming in contact with the cell membranes and dry heat. So let's think about that and work through that. All right, so we know that water conducts very well. Boiling water is a very good way to control microbial growth. Now, if we go back to the continuum, boiling water will not sterilize, but it will get rid of all the protozoans, get rid of a good many viruses, good many bacteria, gets rid of some cysts, but not always, but it will not get rid of endospores. So therefore, it does not sterilize. So boiling water, and you should know this, it's just good information to know, for 100 degrees Celsius, that's 212 Fahrenheit, 100 degrees Celsius, boiling water, if you boil water for 10 minutes, we know time is a factor in getting rid of microorganisms, boiling water for 10 minutes will get rid of most things. So this is a good idea if drinking water is contaminated. They may tell you to boil water to be safe. Just know it won't get rid of everything, and it definitely will not get rid of endospores from that. But it works pretty well. Okay, so that is moist heat and controlling microbial environment with a moist heat. Now, there is another type of moist heat, again, taking water and raising the temperature, and this is called autoclaving. And in this case, we get the water to 121 degrees Celsius. 100 degrees Celsius is the pot on your stove. 121 degrees Celsius for 15 to 20 minutes will get rid of endospores. This will sterilize. So what's the difference from that? Let's go on just to help you understand what happens. If I've got water in the stove and I'm asking and it's boiling, it's going to be 100 degrees Celsius. I cannot get my water on my stove here at my house to be hotter than 100 degrees Celsius. Doesn't matter how long it boils, the temperature, every time I check it with a thermometer, it'll be 100 degrees Celsius. And the reason is that what I'm asking the water to do is change states from a liquid to a gas. The liquid in the water is going to reach a certain temperature, and those molecules in the water are going to move fast enough that they're going to change states of matter and go from a liquid to a gas. And that's going to happen at 100 degrees Celsius. And what that is here in Houston, in sea level, the air pressure that's pressing down on my water molecules and moving them, it's going to take 100 degrees Celsius to be able to get those water molecules moving fast enough to push past the pressure of the air coming down on it. And that's going to be 100 degrees Celsius. Now, I cannot get my water to be any hotter than this because when it hits that point, it's going to change from water to gas, to steam. And that's what's going to happen from that. Now, let's talk about pressure and how that works. If you're living in Denver, if you're living in the Mile High City where you are up high on a mountaintop and you're going to boil water. Does water still boil at 100 degrees Celsius? Take a second, pause, think about it, ask your group, I don't know, think about it. Does water still boil at 100 degrees Celsius on the top of a mountain? Anybody made a cake or something with a mix and you'll look at the directions on the cake box and it'll have high altitude instructions? That's a clue. Things behave differently at a higher altitude. What's going on? I'm at the top of a mountain. I am way up here in Denver. The amount of air pressure pushing down on me at the top of the mountain is different than the amount of air pressure down here at sea level. Down here in Houston, I've got more air pressure pushing down than I do at Denver. I have less air pressure pushing down. There's just less air up there to push down. You can think of it also if you're diving. Who are my diving people who like to scuba dive? If you go down deep underwater, you've got all that water pressure pushing on you. And if you try to rise too rapidly, you get something that bends. That pressure makes a difference. So pressure makes a difference in changing water from a liquid state to a gaseous state. So Denver, Colorado. Less air pressure, it's going to take less heat to be able to overcome that air pressure and change from a liquid state to a gaseous state. Water will boil at a lower temperature than 100 degrees Celsius, which is what we have here at sea level. It's different. So now, if I go back to my PowerPoint slide and we talk about autoclaving, I need my water to get to 121 degrees Celsius. So I need to increase the pressure. By increasing the pressure that is pushing down on the water, then I can let that water temperature get hotter than 100 degrees. So how do we do that? We do this by creating a closed environment. This is an autoclave. This is where we're going to use moist heat. We're going to close off the environment. We're going to increase the pressure in this. And so it's going to take longer for my water to go from a liquid state into a steam state. The water will get hotter than 100 degrees because the pressure pushing down, and for this it's 15 pounds per inch squared to get this, the pressure pushing down is more. All right, autoclaves, because it can get hotter and because we're leaving it for a little longer time, doesn't matter how long you leave this, it's still not going to get rid of stuff, but because we can get it hotter, we can kill more stuff. And we can kill more microorganisms and we can kill those endospores, which gets us to sterilized. And that is good to know. That's our highest level of disinfection. All right, so what do you need to know about the autoclave? You don't need to know all of this stuff other than it just increases the pressure. We do need to know it's a moist heat. We use these in doctor's offices. We use them in dental offices. We use this autoclave in lab to get our sterile media. We put our media in an autoclave to get rid of all the microorganisms. Nail salons, when they go, if you get your nails done and they open that package in front of you, hopefully they do, that package has been autoclaved, and that will remove all of the microorganisms, including endospores. So autoclaving is a very effective way of sterilization using moist heat. Now, as we go through these methods, you do want to keep in mind what will sterilize, because if I'm asking you or giving you a problem, if I'm trying to get something sterile, you need to know the methods that will sterilize. So keep that in mind as you're working through these different physical methods. All right, we use moist heat. We're using steam, moist heat to control microbial growth, and because we can get it hot, an autoclave will sterilize. What's another method of moist heat? Well, that pasteurization. That is also a method of moist heat. We are moving liquids through, we're going to make them very hot and then make them very cold. And this is what we use for our milks and our orange juices and that kind of thing. All right, so I'm going to draw on the board for this. So I'm going back to my handy dandy whatever. How does this work? How do we do that? I'm going to run my liquid through a pipe that is heated. So here's my pipe, here's my liquid running through my pipe. The pipe is heated. I can control how long it's in this pipe, and I know how long it needs to be in this heated pipe to get a liquid to a certain temperature. So it's going to stay at a higher temperature for a period of time. I am then going to move it through a section of pipes that is cold. And so as things move through this cold section of pipes, temperature is going to decrease and I can cool this off. This is pasteurization, taking it up to a high heat for a certain period of time and then following up by a cooling period and bringing it down. Again, this will not sterilize. Sterilizing liquids for food and drinks is going to change the taste. So this will not sterilize. It heats it up and then cools it down. For the most part, let's go through. All right, so back to the PowerPoint. Let me show you the different types of pasteurization. So we know pasteurization because of Louis Pasteur and all his work with the wine. This is what they called historical pasteurization. After he did the work with the wine industry, he kind of fooled around with it because he wanted the wine to taste good, but we didn't want it to spoil with all those bacteria making acids. So they came up with a where they heated the wine to 63 degrees Celsius for 30 minutes and then moved on, to other types of liquids pasteurizing. This is an old type of pasteurization. We don't do this anymore. What we do now is this flash pasteurization. Because we're moving these liquids through these tubes, we can get the temperature up to 72 degrees Celsius and just keep it there for 15 seconds. And then we send it through the cold section of the tube and we can get the temperature to go down. So we let the temperature go up a little bit more, but we're doing it for a shorter period of time. This is typically what they use now. So the milk that you're buying in your grocery store, just regularly off the, out of the refrigerated compartment, is a flash pasteurization, 72 degrees Celsius for 15 seconds. But now I told you it wouldn't sterilize, but what about those organic milks? What about those, those juice boxes that have the milk that you can put in your kids' lunch boxes? Those are in the containers where they're protected from light. They're in the opaque containers. You can't see through them. A lot of your almond milks, your organic milks or something, they're also in those cartons where you can't see through it. And those are the ones that you buy and you look at the expiration date, and like regular milk, you got like maybe 2 weeks or something. Those milks, you might have 3 months on your expiration date before it expires. What's going on with that? What's the difference between the two? Because I told you, even milk in your refrigerator, even if you don't touch it and open it, just regular milk, because it's pasteurized, it leaves some microorganisms, it will still spoil. But my organic milks, I got 3 months before it spoils. That is actually going through a process called ultra high temperature sterilization. It is a type of pasteurization. You get a higher temperature, so you actually are removing all of the microorganisms. If it says ultra high temperature sterilization, you effectively remove all the microorganisms. So then it won't spoil. As long as that seal is not broken, it won't spoil. That's why they can put these expiration dates 3 months, 6 months out, because it shouldn't spoil. It has been pasteurized by this ultra high temperature method. for that. So look on it, look on those products that you see and look for that UHT, and you know that it was treated that way, and that's why you've got that expiration. Now, hint, once you open up the milk, you do not have that 3 months anymore. Once you have opened it up to the environment, potential contamination, bacteria could get in there. Now they say it's good in the refrigerator for 1 week, 10 days maybe. So once you have broken that seal, you don't have that 3 months anymore. So good things are to do is to write down the date of something to open like that, because once you open it, then it's potentially contaminated and it will spoil more quickly. Okay, so ultra high temperature sterilization. These are also, again, higher temperature for less amount of time. These are also why your coffee creamers at your restaurants can be in those little bitty packets and they're room temperature and they're fine. It's because they're treated with this UHT method. Okay, so understand the process of pasteurization, understand the difference. You don't have to get particular and memorize this kind of stuff, but you should understand the process of moist heat from that. Okay, all right, that is moist heat, but let's talk about dry heat. Some things can't get wet, right? If you have something that's fragile, powders, medicine, metals that can't handle the water, they can't handle moist heat, but we still can use heat as a physical method to control microbial growth. But just like your oven, things will cook faster in boiling water than they'll cook in your oven. If I'm cooking my chicken at 350 in my oven versus throwing a chicken breast in boiling water, which one's going to cook faster? It's going to be your chicken in your boiling water because it transfers heat. That water transfers heat faster. It's going to cook faster. So I can use dry heat, and that's good if the substance I'm trying to treat can't handle water, but I'm going to have to do it for a longer period of time. I'm going to have to get a higher temperature for a longer period of time. So we can sterilize this way. If I do it at 171 degrees Celsius, so remember, water boils at 171 degrees Celsius, pretty hot, for an hour, that will sterilize. If I get 160 degrees Celsius, I need to leave it longer, but that will sterilize. Problem is, if it's fragile enough that it can't handle water, a lot of times this is will incinerate these things, or things will catch on fire. So you have to be careful. So they can use dry heat for some things. Our incineration, when we use our Bunsen burners in lab, this is actually a method of dry heat. We're using that metal can handle heat, and we are just burning up anything that may be on there, any bacteria, anything else. This is sterilization. When we talk about trying to get rid of prions, incineration is about the only thing we can do. There is an enzyme, we'll get to that, but incineration, this is an ultimate means of sterilization. It gets rid of everything. And this is what we do to potentially contaminated tissue samples, dressing samples. When we had Ebola here 5 years ago, that was harder to get rid of. They would take the dressings and the tissue sample and undergo incineration with that. So that is a method of dry heat. Okay. Okay, so I'll try to see. Okay, so let's go ahead, and this video might get a little long, but let's finish up the physical methods. A lot of these are things that you'll understand and know and deal with. So let's keep going. Refrigeration and freezing, using temperature to control microbial growth. Absolutely. Refrigeration, we are getting it down to not freezing. 0 degrees Celsius is freezing, that change in the state of matter from liquid water to frozen ice water, but we're not getting it down that far in refrigeration. We're getting it down to about 4 degrees Celsius is typical for your refrigeration. And then freezing is when you get below, when we change that state of matter. When we're talking about refrigeration, what we're doing there is we're just slowing down metabolism. Do you remember our metabolic growth curve? Here, I'll go back and draw it for you. So we talked about bacteria growth over time, and we said that bacteria had an optimum growth temperature. Oh, this was, yeah, temperature's over here. And this is time, and it has an optimum growth temperature. But if we get too much, I am messing this up. Here, let me try this again. We're going to put temperature here. This is the number of microorganisms on this chart, microorganisms. All right, so I've got a temperature that's my optimum growth temperature. I'm going to get the most number of microorganisms growing at this optimum growth temperature. If I get it too high, remember it falls off. I'm not going to get a lot of growth. If I get it too low, it's also going to fall off. But we talked about this in chapter 6 with microbial growth, and we said that this side, where it gets cold, we just decrease metabolism. We slow down the rate of reactions. So metabolism is slowing down here. Heat, we're denaturing stuff. We're killing stuff. We're killing stuff when we're adding the heat. Denaturing proteins, you can't come back from that. But this side of the temperature, we're just slowing down metabolism. So that's how refrigeration works. All we're doing is we're slowing down the rate of growth. So you put that pasteurized milk in there, and microorganisms are there, and they can replicate, but they're doing it slowly. So you got 2 weeks. If it's not pasteurized, you got maybe a day before those microorganisms will grow. There were more microorganisms to start with. So metabolism is still slowed down, but because we started with more with unpasteurized milk, that's the differences there. So refrigeration is just slowing down metabolism from that. When we get to freezing, freezing actually changes the state of water. So we know microorganisms need water to grow, right? I spent that whole time on metabolism. We need water to undergo cellular respiration. If we do not have water, if all of our water is ice, no growth. So that is going to stop the growth of most pathogens. So I use freezing. Again, you can keep things in the refrigerator, leftovers, maybe a week or so. In the freezer, you got longer. You've got 3 months, 6 months, maybe a year, and you can still come back from that. It's because microorganisms' growth has stopped. Long enough time usually kills microorganisms. If it's a short time, it may inhibit the growth, but it may still resume growth once you take it out of the freezer. If you leave it frozen long enough, you can kill more things. And when my kids are little in the spring, I would take a bird's nest that we found in the wintertime and I'd stick it in the freezer. And after a couple of months in the freezer, when it came spring, I could bring that back out and the kids could touch and pull apart the bird's nest and get hands-on with the bird's nest because I killed anything that might be growing in there. So it's a good way to control microbial growth in the environment. Just physically, no chemicals needed for that. All right, so freezing. Let me talk a little bit about some different types of freezing. When I just stick something in my freezer at home, it's going to take some time before the material gets below 0 degrees Celsius. This is considered a slow freezing method. And this works, but as it's freezing slowly and that water is turning into crystals, the ice crystals are actually very sharp and they can damage cell membranes. So those ice crystals the water slowly turning into ice right next to your cell membrane are actually poking holes in your bacteria cell membrane. That's why they're dying, because you're poking enough holes in the bacteria cell membrane. So this is a slow method from that, and it usually is good for stopping things growing. So that's good. But sometimes we use freezing and we don't want to kill microorganisms or we don't want to kill cells. We can use a quick freezing technique and stop that water from making those crystals and damaging cell membranes. They still freeze, but because they're freezing so quickly, it's a different structure and they don't damage the cells. Still some damage, but not as much as it would. Because we know freezing my chicken breast out of the freezer after 3 months or 6 months, it's not going to taste the same as when I did it fresh. There's damage. Because of those ice crystals, you're damaging proteins, you're damaging cell membranes. Okay, so let's talk about fast freezing. So say I want to preserve cell membranes. If it's a cell line in a hospital, say you've got a cancer patient and they're being treated and it's going to affect their reproductive ability. And so they say they want to freeze some cells, freezing cells in the ovaries or fertilized membranes or freezing sperm. We want that to survive freezing. We want our in vitro fertilization, where you fertilize embryos and then you take the embryos and freeze them for future attempts at implantation. we want that to survive. When we quick freeze things, we can make these cells, they're not as damaged as much and they can survive. So we use liquid nitrogen to quick freeze. Liquid nitrogen is for that. We use them for cell lines. We use it if we're shipping bacteria for our labs across we need to order more E. coli or something. We ran out. We had a mistake. We couldn't go into our lab for 3 months and all of our E. coli died. We didn't have anything frozen. We can order it and they'll ship it to us that way. That is frozen in liquid nitrogen, so we can do that. The thing you know about is, anybody gone to that, what is it, Cold Zero? What is that ice cream place over in the Vintage? You know what I'm talking about. You'd tell me if you were talking to me about it. But they use the liquid nitrogen to fast freeze the ice cream, and they say it tastes better. It's because they're not damaging those cell membranes as much. So that's what's going on there. All right, so freezing will kill more things. Slow freezing, if you're trying to kill stuff, slow freezing is the way to go. It will not sterilize. Some things can survive, and there are some pathogens that can live in colder temperatures. Listeria, most pathogens, again, freezing and refrigeration works really well. Listeria is an exception. It will actually survive lower than 0 degrees Celsius. And that's why we couldn't have Bluebell for a whole summer, because their ice cream plants got contaminated with listeria, and they had to do the proper disinfection protocols from that. Okay, let's go back to the PowerPoint. What's next? Let's make our way through. Okay, the next section is desiccation and lyophilization. Desiccation is drying something out. So this is food dehydrators. This is, oh, tell me this. Which would you rather, which will you be able to keep longer? Raisins or grapes? Raisins are just grapes that have been dehydrated and all the water has been removed. We know microorganisms need water. So if we remove the water, then we can keep these things longer. So controlling microbial growth in the environment, removing the water does a very good job with that. And that's how raisins work. Be careful, molds will grow on bacteria. Maybe the molds will grow on dried fruit. Maybe bacteria won't, but you do need to be careful about molds. Okay, desiccation. After desiccation is lyophilization, and this is the, have anybody been to NASA and gotten like the ice cream, the astronaut ice cream? This is where we use freezing and removing the water. This is called lyophilization. Go ahead and say that. That's a hard one. Lyophilization. And this is an example of that. If anybody backpacks or does any of this stuff, this is some lyophilized, I wish you could feel it. It's very light. This is lyophilized green beans. They removed all the water and then they flash froze it, and then they packaged it in this, and backpackers can take this, it's very light, you pack it in there, and open up the package and pour your boiling water right into the package, and it reconstitutes the green beans. So my husband got really into backpacking for a while, so we have a bunch of this over there, so we're not going to starve in my household, even in quarantine. And this is called lyophilization, freeze drying. And that's a method of controlling microbial growth. And in fact, on the back, it doesn't have an expiration date. It has a manufactured date, because theoretically this can last forever. It was manufactured in 2014, and it's still good now. If I poured water in this, hot water, reconstitute it, then it would be just fine for that. So controlling microbial growth does a very good job. All right, so that's lyophilization. Desiccation, lyophilization. What's next? Physical methods to control microbial growth. Physical methods, filtration. I can actually take liquids with bacteria, pass them through a filter, and if the pores are small enough, the bacteria can't go through. So this is really good for different drugs, different solutions, vaccines that you can't use other types of physical methods to control microbial growth and you don't want to add chemicals. You can pass it through a filter. The smaller the pore size, the more you get rid of. You can get pores that will get rid of bacteria. You can get pores that are small enough that you can get rid of viruses, but they're more expensive from that. I actually have an example of that one too. Again, thanks to my husband and all his prepping and backpacking adventures. This is a product called the Life Straw, and it is a filtration method of getting clean drinking water. And so presumably it's got the filter in here, and you can actually just put it in the pond and take care and just drink the water straight out of that. And because of this filter, it will remove any protozoan cysts, any bacteria. Now this one won't get rid of a lot of viruses, but in North America, ponds and drinking water, viruses aren't an issue. Well, that's not true. Not as big of an issue. Now, different other countries have more problem with viruses in the water, but our country's not quite as bad. But either way, I can boil it to get rid of the viruses, and then I can use this and get rid of protozoan cysts. I can use both of those methods to make it very safe. But this actually works pretty well as well. So you can check out this company and see how that works. So filtration. How do we use filtration? They use it in treating drinking water in water treatment plants, sewage to contaminate from that. And again, we already said smaller ones to do drugs and other types of things. Okay, so another physical way we can control microbial growth. If I add enough salt and enough sugar to the outside of a cell, then I'm going to alter the water, and that will control microbial growth. This is using osmotic pressure to inhibit microbial growth. So I am a sailor, and I caught some fish, and now I'm going to just cover it in salt. And this, you need to go back to what you remember from hypotonic and hypertonic. Back to my little whiteboard here. And so if I've got a cell, here is a cell. So I've got a fish. That's my fish. And this is my bacteria on my fish. This is not drawn to scale, but I got bacteria. We'll draw a little bit better. I got bacteria living on my fish. My fish is now dead, so I'm going to get more and more bacteria living on my fish. What I can do is I can add a solute to the environment. I can cover this bacteria, the fish, and any bacteria that is growing on it with salt. Now, when I talk about that, what's going to happen to the water? Going back to hypertonic, hypotonic. Hypertonic, if I have more solutes outside my bacteria cell, my water is going to leave my bacteria cell and go outside my cell. We know microorganisms need water. All the water is leaving that bacteria because there's salt all around it. This is a way we can physically alter the environment to control microbial growth. This is how fishermen would catch fish and then be able to survive off that for a long period of time and preserve that on the boats. Jams and jellies work the same way. They just use sugar as the solutes for that. Now, with our jams and jellies, and you know this, your jams and jellies will last a very, very long time in your refrigerator. Molds will still grow. They are less susceptible to this method of controlling microbial growth. So watch out for the moldy jams and jellies. All right, that's using osmotic pressure to physically alter the environment and control microbial growth. All right, radiation. I can alter my environment by increasing the radiation in my environment. There's ionizing radiation and non-ionizing radiation. Let me talk about the ionizing radiation first. This is an effective way to control microbial growth. Let's talk about gamma rays, which is a type of ionizing radiation, and our strawberries. Now, I don't know about you, but I go to the grocery store and I buy strawberries like this, and after one day, I swear, they look like this. Maybe 2 days max. They look like this. So what is a way, and why do they do this? Well, that's bacteria. There's bacteria on here. There's fungus on here. That is, the microorganisms are growing. They're using the strawberries as a food source, and my strawberries are rotting. This is a problem. So I like my strawberries, but I don't want all the microorganisms that come with them. If I treat my strawberries, or if the companies treat the strawberries with these gamma rays, which are approved by a lot of these organizations, Food and Drug Administration, U.S. Department of Agriculture, Environmental Protection Agency, the World Health Organization, all of these have approved gamma rays to use to control microbial growth for meats, fruits, vegetables, spices. We can do this. We pass these gamma rays through the fruit. The radiation in this type of thing does not build up in the fruit. So it's passed through the fruit. Any microorganisms have lost their reproductive ability. They will not grow. Now, any seeds in the strawberry have also lost reproductive ability, but I'm not interested in planting the seeds. I'm interested in eating the strawberries. So passing the microorganisms, passing the gamma rays through these microorganisms will get rid of anything growing on there. And if it's not growing on there, then it's not going to desiccate. And my strawberries aren't going to look like mush. Now, again, got it out in the environment. Can't keep it forever. It'll pick up more microorganisms. But I can keep my strawberries for much longer than the few days that it seems to work. So gamma rays is an effective means of controlling microbial growth, and it's proven safe. We've been using this for decades. We've got lots of different studies on this, following long-term studies with growth. It doesn't, unlike other forms of radiation, it doesn't stay in the strawberries. Strawberries, it gets passed through, and then there's no residual radiation. So you consuming the strawberries is perfectly healthy. Okay, you've probably not heard of this. If a fruit or vegetable or a meat has used this method of microbial growth, it does have to have this label on this. You've probably not seen that, and it's because in this country, we're not big on this. We do have to label it. It says that it's been radiated. And public perception, if I told you were eating radiated strawberries, your first gut reaction is usually negative. So in this country, we're not big on this. We're very cautious about the use of radiation. We're very fearful of it, and we're worried about it. So whenever this label goes on, things don't sell as well. So if this label goes on, it doesn't sell as well, then people aren't going to use it. So we don't use this a lot in this country. You probably have already eaten something that's been irradiated, because our spices, if we get this from other countries, we don't have to put the label on it if it's a certain percentage. So a lot of your mixed spices, they'll use this in other countries and then they get shipped to here. So you're probably eating irradiated food, you just don't know it. But here we don't use this as much. So it will kill anything, kills microorganisms, their larvae, their eggs, and therefore lengthens the shelf life. We do use radiation to sterilize dental and medical supplies, so it is an effective physical way we can control microbial growth. Now that is ionizing radiation. We can also use non-ionizing radiation, and you've heard some things about that even with this COVID-19. They're like, put it outside, let the sunlight get on this UV light. If you leave it enough time, UV light can control microbial growth. So that is good. What it does is it damages DNA. These thymines, instead of binding to the other strand like they're supposed to, they bind to each other. So it creates these thymine dimers. But this is also a way that works to control microbial growth. Now, what you need to know, just like when you're going outside for your suntan, UV light doesn't penetrate very well. So clothing, hats, umbrellas, shade, we use that to protect ourselves from UV light. Same thing. If I'm trying to use UV light to disinfect drinking water, and they do use this in water treatment plants, then it's got to be able to penetrate. So it's good for disinfecting water. It's good for disinfecting air. They actually use this in hospitals. There's a machine that they can roll into a patient's room and use UV light to get rid of everything there, and that works well for surfaces that the UV light can contact. Anything below the surface is not going to come in contact. It doesn't penetrate through surfaces. So it's great for air, it's great for water from that. We get a lot of things. My husband wants to buy one of these so bad. The little things that you can put your phone in and get UV light to do that. Those are hot right now, especially with this COVID thing. I think they sold out like completely. Are they a good idea? Are they a bad idea? They'll work, but so will a little alcohol wipe. And then remember, it will not penetrate. So if here's your phone, and you've got organic matter, dirts, oils, anything from your hands on your phone, you stick it in that UV light, it may not penetrate. You may not be doing what you think you're doing. So yes, it's a good method. Just know the pros and cons from that. All right, so UV light is a way to physically alter your environment to control microbial growth. Okay, so we made it through all the physical methods. This table is in your book. This is a good table that's got all the methods in there. It's got your conditions. Don't need to memorize this table, but a couple of them I highlighted. I think you should know how long you should boil water for it to be as safe as possible. So don't memorize this, but it is a good method to compare these, and this column. How do we use it? Because on your exams, I'm not going to be interested in you parroting back information. I'm going to give you a scenario, and I want you to be able to pick the best disinfection method. Know the pros and cons, know when this is good and when it's not good. So that's what's going to do for that. Okay, that's probably a long video, but we got through physical methods of microbial control, and the next video we'll talk about how we can use chemicals to control microbial growth in the environment. All right, stay with me. Thanks. Bye.